Hello friends, myself Dr. Saurabh Pandey, and today we will discuss about the RNA interference and virus-induced gene silencing. This is an important topic for different exams like KSRP, CSIR, TVT, UPSC and NEET. So coming to the lecture, what we will learn in this lecture, we will know about the gene silencing, what it is and what are the different types of gene silencing. Then we will know about the RNAi interference, important components of RNAi interference, its mechanism and processes, then its application, then the advancement of the gene silencing, that is virus-induced gene silencing used for the characterization of genes. We will learn about its processes and players, then its applications, and I will give you some questions for the next lecture. Okay. So what is gene silencing? So as you know the central dogma of life, any gene that has to function, it has to transcribe and translate into the protein. So if any process in between get disturbed, it is called as gene silencing. Normally the silencing is happening at the transcriptional level, that is for the conversion of DNA to RNA, or post-transcriptional level after the conversion of DNA to RNA and before the protein production. So therefore gene silencing is the regulation of a gene in order to reduce its expression. This is important. Normally in gene silencing we are not switching off the total expression of any gene, we are just reducing its expression. Okay, so as I already told, it can happen at transcriptional or post-transcriptional level. In transcriptional gene silencing, mostly the hypermethylated regions in the promoter are responsible for the gene silencing. Okay, whereas in post-transcriptional gene silencing, hypermethylated regions of the coding part is responsible for the gene silencing. So one of the main gene silencing tool is RNA interference in the living system. So what is RNA interference? As I already told, in the central dogma, for any gene to perform its function, it has to first transcribe into RNA, then it has to translate into protein. So in RNA interference, what is happening? A specific sequence of genes get silenced or knocked down by the production of short duplex strands. Okay, so this has different forms like we already discussed, post-transcriptional gene silencing and transcriptional gene silencing. So the basic requirement of RNA interference is a target RNA from which the double-stranded RNA sequence get formed, and it will get suppressed by the activity of RNAi machinery. So here you can see one term, knockdown. There are two or three terms. The one is knockdown. In knockdown we will reduce the expression. In knockout we will remove the gene responsible for the expression, therefore there will be no expression. Okay. So there are some synonyms of RNAi. The plant biologist uses post-transcriptional gene silencing and co-suppression. The animal biologist called it as RNA interference, and the mycologist, they called RNA interference as queering. So coming to the timeline of RNAi, it started with the discovery of post-transcriptional gene silencing in the overexpression plants of tobacco in 1990. Further, in 1992, quelling was discovered in Neurospora. The same thing, transcriptional, post-transcriptional gene silencing in Neurospora fungus, uh, called as quelling in fungus. In 1998, two scientists, Fire and Mello, discovered this phenomena as RNAi in nematode Caenorhabditis elegans, and they got Nobel Prize for that. After that, this RNAi was discovered in many other organisms like yeast, Drosophila plants, trypanosomes, etc. By the 2001, there were many successful examples of siRNA or RNAi mediated uh, reduction in expression of mammalian cells genes. Then by 2002, recombinant components of the RNAi were generated. In 2002 itself, science named RNAi as a technology of the year, and by 2003, it was the billion dollar industry. So, how the idea of RNAi came? First of all, in 1928, Wingard performed an experiment on tobacco plant. So the infected tobacco plants with the virus shows very strange phenomena of infection symptoms. So after the virus infection, tobacco ring spot virus infection, only the lower leaves were infected, but somehow the upper leaves, all the upper leaves showing the recovery and they were healthy. So he has no idea why this is happening. It was because of the post-transcriptional gene silencing that is chopping off the virus RNA and giving the defense to the younger leaves. So the early demonstration of RNAi was happened when scientists tried to overexpress the chalcone synthase gene in Putinnia. As you know, chalcone synthase comes at the anthocyanin synthesis pathway and it gives a particular color. So wild type petunia produces purple type of anthocyanin pigments. So scientists tried that if we overexpress this gene, we will get more pigmented flower. But to their surprise, what they saw, instead of more color for chalcone synthase, it resulted into variegated type of phenotypes. So as you can see in this picture, there were so many different types of combination of color in petunia plants. So why this was happening? Uh, it was happening because before the RNAi was known, the simple way of expression was if mRNA is attached to the ribosome, the protein synthesis will take place, and if there is no mRNA is available for attaching to the ribosome, there will be no protein synthesis. So the hypothesis was that if sense RNA production if sense RNA production will be enhanced, the pigmentation will be enhanced. So in normal case, endogenous gene will produce mRNA and it will get translated and we will get purple color. And the hypothesis was if we express the transgene, the extra protein will be translated and we will get extra color. But what happened? The transgene expression resulted into the RNAi phenomena where sense and antisense RNA were formed duplexes and they lost the color. But surprisingly, what happened? Scientists thought that only antisense RNA will form the double stranded RNA complex and get chopped off. But to their surprise, sense gene constructs also inhibit the pigment production. So why this was happening? This was discovered by the Fire and Mello, as I already told, in Sinorhabditis elegans. So any introduction of long double-stranded RNA resulted into the degradation of corresponding mRNA. That phenomena they coined the term as RNAi. So simply what is happening, if any double-stranded RNA is present in the host cell, the host recognized it as a foreign molecule or abnormal molecule. So there will be one enzyme that will cut these double-stranded RNAs into smaller RNAs. That is known as small interfering RNA. These small RNAs will get associated with protein complexes and get chopped off the mRNA that is targeted. This is the simple phenomena of RNA silencing. We will come to its component now. The main component of RNAi machinery is different enzymes and proteins like dicer enzyme. It is a protein that is having RNase activity, so it will degrade the double-stranded RNAs into smaller RNAs known as small interfering RNAs. Then there is protein complexes, that is RNA-induced silencing complex, also known as RISC complex, which is a large protein complex that is activated in the presence of smaller RNAs produced by the dicer enzyme, as we already seen. So the core of RNA silencing are dicers and Argonautes protein. Dicers, as we already discussed, in animal systems dicer proteins are there, but in plant systems dicer-like proteins are there to chop off the double-stranded RNAs. These chopped off proteins were subsequently associated with the Argonaute protein that takes it to the silencing complex for further processing. So coming to the dicer and dicer-like protein, it is most important protein because it identifies the double-stranded RNA and chop chopping of action was performed by the dicer. It is known as dicer in animal, but dicer-like protein or DCL in plants. Okay, and it usually generates 21 to 25 nucleotide fragments. So importantly, DCL1 and DCL4, if they are in action, they will generate 21 nucleotide double-stranded RNA, whereas DCL2 generate 22 nucleotide double-stranded RNA and DCL3 generates 24 nucleotide double-stranded RNA. So based upon different uh, participation of different DCLs, they will perform different types of chopping and produces different length double-stranded RNAs. Okay, so how the dicer term came? Because it is chopping of the RNA as a cook which dices the vegetable. And the term dicer was coined by Emily Bernstein in 2001. How dicer is performing this action? Because it is a endonuclease and having the RNase activity. The important question that can come from dicers, how many domains dicers have? Dicers normally have two domains, and one domain is RNase3 domain, which is having RNase activity, and the other one is the patch domain, which decides the uh, binding of the protein with the double-stranded RNA molecules for the production of siRNAs. The important thing of dicer chopping is after the chopping of double-stranded RNA, the mature miRNA will present with the 2 nucleotide overhang at the 3' end. Okay, this is important. This is important because after after the kinase activity, after the chopping off, this will act as signal, the 2 nucleotide overhang. Okay, that will facilitate to the RNA-induced silencing complex by the Argonaute protein. Okay, and leads to the degradation of mRNA. So coming to the Argonaute proteins, it binds to the chopped off small RNAs generated by the dicer or dicer-like protein. It is also an important component of RNAi machinery, and it has a patch domain for binding the 2 base 3' overhang that we already discussed, produced by the dicer proteins. Okay, it also has a PV domain which forms RNA's H-like fold, which is required for the guided hydrolysis of single-stranded RNA. This is very important. The question that can be asked, which domain is responsible for the guided degradation of single-stranded RNA? Okay, there are different types of Argonaute proteins like AGO1, 2, 4, 7 and 10, which performs the slicer activity. So how the name of Argonaute came? Argonaute resembles one octopus, Argonauta argo, in the shape. Therefore, it has given the name of Argonaute protein. The last important component of RNAi machinery is RNA-induced silencing complex. It is a protein complex with 2 RNA binding proteins. These complex having RNA or DNA helicase activity as well as translation initiation. The important thing is the RNA-induced silencing complex also has RNA-dependent RNA polymerase activity. What it is? They require RNA primer for the synthesis of RNA. Okay, and it is a transmembrane protein. After the recognition of double-stranded RNA, it gets chopped off by dicer-like protein. Argonaute protein binds the chopped off siRNAs, and they will associate with the RNA-induced silencing complex for further action. Here, the antisense stand is very important because only it can define the degradation specificity of RNAi. So these were the main component of RNAi. But what are different small RNAs that were generated after the chopping of dicer or DCL-like proteins? So what are small RNAs? Small RNAs are a pool of 21 to 24 nucleotide RNAs that generally functions in gene silencing. Okay, so as we discussed, these RNAs contributes to the post-transcriptional gene silencing as well as transcriptional gene silencing. Okay, the post-transcriptional gene silencing will affect the mRNA stability, therefore the translation process. So this will known as the translation inhibition, whereas the small RNAs which contributes to gene silencing by the epigenetic modification to the chromatins like histone modification, DNA methylation, etc. They will cause the transcriptional gene silencing. These two things are very important. One is the post-transcriptional gene silencing. Okay, the other is transcriptional gene silencing. So different components will take place. Different components will take place for both these events. One is the siRNA. siRNA, as we already discussed, after the chopping of DCL, these are the 21 to 22 nucleotide in length, and they are having specifically 2 nucleotide 3' overhang for recognition to the Argonaute protein. It will further processed for the RNA interference. The other type of small RNAs are the microRNAs. These are the small RNAs that are produced by the endogenous genes of host, and they somewhat perform the similar action of that we already seen for small interfering RNA, siRNA. So the miRNA gene will translate to the double stranded RNA complex known as RNAi. It will recognized by the DCL proteins that will get chopped off, where Argonaute protein will bind, and it will associate it to the RNA-induced silencing complex, and the targeted mRNA degradation will happen. So in overall view, how RNA silencing works, it can work by both these RNAs, siRNA mediated silencing and miRNA mediated silencing. siRNA mediated silencing normally performed by the post-transcriptional as well as transcriptional level gene silencing, where uh, double-stranded foreign RNA molecule will recognized by the dicer-like protein. It will be degraded by the Argonaute protein. Argonaute will take it to the RISC complex, where it will further get processed. In miRNA mediated silencing, what is happening? The miRNA will bind to the uh, mRNA, and translational repression will happen, also known as post-transcriptional gene silencing. Okay, so these are the basic RNA silencing uh, phenomena. So coming to the mechanism of RNAi will be completed in three different phases. The first one is initiation phase, the second one is maintenance phase, and the third and important one is signal amplification and spreading phase. So in initiation phase, double-stranded RNA will be synthesized, okay, and that will get chopped off to form the small interfering RNAs. In maintenance phase, these small interfering RNAs will be associated with the RISC complex, where it guides the nuclease activity and degradation of target mRNA. So whatever the siRNAs are produced, they will associate with RISC complex and uh, inhibiting the expression of mRNA. Then the important phase is signal amplification, where the siRNA act as the promoter of generating more double-stranded RNA, which will generate more siRNA. Therefore, signal amplification will happen. So this is the initiation step, where single-stranded RNA from viral RNA or hairpin RNA will be converted to the double-stranded RNA in the cell, and these will be recognized by the dicer protein, and by the energy consumption they will be converted into the small interfering RNA. These small interfering RNA will then get phosphorylated by the kinase uh, enzyme, and after that, by the activity of RNA-dependent RNA polymerase, this different SIR, more siRNAs will be produced from the double-stranded RNA template. Coming to the effector step, these siRNA will then bind to the protein complex that is known as RNA-induced silencing complex, where only because I already told it has helicase activity, which will be responsible for uh, unwinding of the double stranded RNA strand. And as I already told, the antisense strand is important, so only antisense strand will remain here, and the other strand will get degraded. So this strand, antisense strand, will then uh, scan for the target mRNA, where complementary uh, binding will happen, and the RISC complex will degrade that mRNA. Okay, these are the effector steps, where the actual silencing is happening through the RISC complex, and the last phase, as was told, was the amplification or spreading. So in initiation phase, the double stranded RNAs were created from the single stranded RNAs recognized by the dicer, and it produces different length of siRNAs. These siRNAs will then bind to the RISC complex. RISC complex then scan. RISC complex then leave out one strand, and only antisense strand will remain with the RISC complex. It will scan the whole genome and degrades the target mRNA. Okay, or it can bind to the targeted mRNA, where RNA-dependent RNA polymerase activity will amplify this signal, and more double-stranded RNA will be created, and more siRNA will be created, and spreading of these signals will happen, and more RNA silencing will be observed. So we already discussed this thing, that siRNA mediated post-transcriptional gene silencing is happening, and miRNA mediated post-transcriptional gene silencing is happening in plants. So what are the basic difference? Basic difference is the siRNA mediated post-transcriptional gene silencing is happening from the outside genetic material or RNA that is coming from the outside foreign. But because plant are having their own miRNAs, so they will produce transcribed transcribed miRNAs with the hairpin loop that is recognized by the dicer, and they will produce the small interfering RNA duplexes. So the process remains same. The hairpin loop or double-stranded RNAs will be recognized by the dicer. Dicer will then binds to them and perform the chopping of action, and with the 3' overhang, siRNA will be generated. These will be recognized by the Argonaute protein, and this protein will target it to the RISC complex. At the RISC complex, only one strand will remain, that is antisense strand, that will go through the genome and scan for the complementarity, and where the complementarity happens, that will be the target mRNA, and it will be chopped off. So there are two important things for RNA silencing. RNA silencing has to happen at the short distance, or extensive silencing happens with the long distance movement. For the short distance movement, mostly 24 nucleotide small interfering RNA are important, and for the extensive silencing movement, the 21 nucleotide siRNAs are important, because it will trigger the de novo synthesis of more double-stranded RNA, and signal amplification will happen, and it can spread over the 10 to 15 cells. So what are the different roles of RNAi? The first and important role of RNAi is immunity to plant. So this system basically developed as the immune system, where any foreign genetic material enters into the cell, it will recognize, it will leads to the formation of double-stranded RNAs, and that will recognized by the RNAi machinery, and it will get chopped off, and so it provides the natural immunity to the plants against different viruses, transposons, or mobile genetic elements that shows the abnormal structures, that is double-stranded structure. So what are different RNAi application? We can genome-wide screen different genes, as done in C. elegans for 16,757 genes. Then there are potential therapeutic use, like antiviral drugs can be created from the RNAi. If we know the target gene, we can produce the drugs. Anti-tumor drugs can also be produced from RNAi applications. Then We can protect the organism's genome from different invading transposones or viruses. Then gene silencing also is a immune system that protects from infectious DNA elements. As a tool, it protects from the virus infection. It secures genome stability by keeping mobile genetic elements known as transposons silent, and the different RNA-like mechanisms represses the protein synthesis and helps in the regulation of development of organisms and regulation of different genes. It is the RNAi also important for keeping chromatin condensed and suppress the transcription at all the time. So it is important regulatory mechanism also. And for gene characterization, we can use RNAi as a tool. Gene characterization mean either we can, if we have to know the function of any gene, we can overexpress that gene. Okay, so like for chalcone synthase, as we already discussed, if we overexpress the gene, there will be more production of the same protein. That is the hypothesis. And if we uh, somehow silence or knock out that gene, so we can know that gene is important for the function of a particular pathway or a particular protein. Like for in petunia, if we silence the chalcone synthase, we will not get any pigments. Okay, so these are the two strategies for gene characterization. So silencing strategies will be performed through the RNA. So there were various examples of RNA interference in plant systems, as you can see in this slide. So it is used in different plants for different purposes. The most important examples are for defense against viruses in banana, in barley and rice, and in other crops, as well as it is also used for enhancing the shelf life in tomato. That question is asked so many times. Okay, so you have to know the gene. ACC synthase was the gene, and that you have to explore. Okay, it is also used for flower color identity modification in different plants, as we have already seen for the chalcone synthase gene. It is known as antisense technology also because antisense is a, there are two strands of DNA synthesis. Like one is sense strand, the other one is antisense strand. From antisense strand only mRNA production will be happening. So coming to the virus-induced silencing, it is the application of or taking the clue from the RNAi and designing the tool for gene silencing strategy. So what is virus-induced gene silencing? So as name suggests, we already discussed the gene silencing. It is happening through the siRNAs, small interfering RNA. So if any virus is inducing such type of gene silencing, it is known as virus-induced silencing. So what we are doing here, we are silencing an endogenous plant gene by the use of viral vectors. So we are confusing the host system by the use of viral vectors. So if we integrate the gene which we want to study with the viral vector, the host will recognize it as the foreign gene and it will perform the silencing action on the gene. So we can characterize that gene in that sense. It is the reverse genetic tools, as we know, because it is establishing a direct link between the biochemical function of a gene product, that is a protein, and its role in vivo systems. Okay, it is a transitional system for characterization of any gene, whereas RNAi were the permanent type of solution. Like we have to create the transgenic. In this virus-induced gene silencing, we don't need to create the transgenics. Okay, and VIGS in plants also takes place if there is any sequence similarity between the virus and or any transgene or any endogenous nuclear gene. Okay, so any sequence similarity between the virus that is recognized by the host, or if we are overexpressing a transgene, if it is recognized as a foreign gene, that will also get silenced, or any endogenous nuclear gene is there which is recognized by the plant as a foreign gene. Therefore, gene silencing can be activated by the this phenomenon. So in this virus-induced silencing, we are using viral vectors for our targeted gene silencing. Okay, there are different viruses which can modified for the silencing of different genes in a sequence-specific manner. And as you know, monocots are different types of viruses infecting them, and as well as dicot. So based upon that, we can modify different viruses for the vector purposes and characterize any genes. And how we perform the inoculations of these viral vectors, we can perform with like simple agro infiltration, because agrobacterium mediated silencing can be happening, and in vitro transcriptions. So what is happening inside the cells? The viral vector having their own genome. So the virus encoded RNA dependent RNA polymerase will generate the viral double stranded RNA that will recognized by the host machinery. Their DCL will act, and the same process will be repeated for up to the silencing of the gene. So in practical, how we are doing this? First, we have to clone the gene of interest in a viral vector. So what is gene of interest? Which any gene which is subject to a study. So we will take the unique sequence of that gene and clone 200 to 300 base pair unique sequence and clone it into the viral vector. Then the cloned thing will be agro-inoculated into the plants. If the agro-inoculation is completed, then the virus will start producing its RNA. These RNA will generate different types of double-stranded RNA, and this will be recognized by the dicer protein, and different siRNAs will be found, and gene silencing will be happening. So if we are inoculating here, we can see the symptoms at the upper portions of leaf. Okay, because virus will move from phloem, so it will move upwards, and suppose this gene silencing was happening for the green color of the plant. So if we silence the chlorophyll synthesis gene, so you can see the bleaching symptoms at the upper portion of the plant. So in summary, this is RNA mediated gene silencing tool. Okay, that is basically derived, we have taken cues from the plant defense to devise this strategy. Here siRNA will interfere with the viral replication. Okay, therefore silencing our targeted gene. These siRNA act systematically. Systematically means they will move from one portion to another during the process of plant recovery and resistance, and therefore we were seeing the symptoms at the upper portion because the virus is moving from this portion to the upper portion systematically. Systematically means spreading basically. Okay, and most viruses produces different suppressor to overcome this phenomena in the plant. Then how this is happening. This is the antiviral RNA silencing that is happening in plants. So viral RNAs will be recognized due to their hair loop formation or double stranded formation. Therefore, they will recognize by the DCL, different DCLs 2, 3 or 4, that will result into formation of different length siRNAs that will then uh, carried out to the RNA-induced silencing complex by AGO1 or 7, where it will uh, leads to the translation inhibition of these targeted genes. So basically, viruses produces different kinds of suppressor to overcome this machinery of the host plants. So these are known as virus silencing suppressors. So you don't need to remember these all things. You just need to remember that viruses produces different proteins like P6, P14, P38, SC-Pro, etc. that will inhibit different steps of RNA silencing machinery that is causing the virus-induced gene silencing. So P6, P14, P38 will uh, try to inhibit the DCL2, 3, 4 step by interacting with these proteins, whereas V2, 2B will interacting with the RNA-dependent RNA polymerase, so there will be no synthesis of double-stranded RNA. Then SC-Pro and other proteins will interact with AGO1 and 7, and the P1 protein also interacts with the RISC complex for basically overcoming the RNA silencing machinery of the plant. Okay, so these are different regulations of the Argonaute protein that can be mediated by the viral RNA silencing suppressor. So simple thing, if virus infection is happening, uh, the DCL will generate the virus-induced silencing, virus-induced siRNAs. These siRNAs will get recognized by the RISC complex taken by the AGO1 and 7, and it will leads to the viral RNA targeting. Okay, but different viral RNA silencing suppressor can inhibit this process. So this is the basic example of how virus-induced gene silencing happens in the plants. So what are the major requirements for virus-induced gene silencing? As we already discussed, we need a viral vector for confusing the host to initiate its gene silencing machinery. Then plant should be feasible for agrobacterium infection, because we have to introduce the viral vectors into the plants by agrobacterium inoculation. Then the target gene silencing needs a particular region of the gene that is unique to that particular gene. Okay, this is major step. So we select usually 200 to 300 base pair or 150 to 300 base pair unique sequence that we will clone in the virus vector and perform the experiment. So how the experimental look of VIGS will take place? We will clone the small fragment, like I already told, 200 to 500 base pair, that has to be unique for that gene. Otherwise, other genes will also get silenced, and you cannot know whether the gene function which is inhibited at that time, it is because of the it is because of your gene or some other gene. So it has to be unique. Okay, that sequence. So after cloning, we will transfect the plant, basically infect the plant by agrobacterium mediated inoculation. Then the double-stranded RNA will form. The basically virus will replicate inside the plant. Viral vector will replicate inside the plant, and double-stranded RNAs will form that will trigger the RNA silencing machinery, because double-stranded RNA will be seen as the unusual RNA, that they will be recognized by the dicer-like enzymes, and that will produce the siRNA. These siRNAs will get amplified, and virus-induced gene silencing will happening. So this is basic one example. There are tobacco rattle virus which can be used for the silencing of the gene. So we cloned the gene in tobacco rattle virus. This is the agrobacterium plate of tobacco rattle virus, where we have cloned the gene for phytoin desaturase. Phytoin desaturase basically takes part into the chlorophyll synthesis. So we mix the growth of this plant, growth of the agrobacterium, sorry, not plant, growth of the agrobacterium, and inject it into the plant, and that will amplify the signal. And as you can see in this example, this gene was responsible for the synthesis of the chlorophyll or chlorophyll color. So if we silence that gene, it will show the bleaching symptom or whitening of the plant. So basically these are the vectors. They have, because it is a agrobacterium mediated transformation, so it will have left border as well as right border. Then it has to have the RNA dependent RNA polymerase activity for amplification of the signal, and post transcriptional gene silencing will happen, and you can see the systematic progression of the bleaching symptoms. So this is basic one example of virus induced gene silencing. So what are the advantages? One advantage I already told, that it is the easiest method because we just have to inoculate the plant with agrobacterium. The cloned viral vector in agrobacterium we can directly inoculate. It is a cost-effective method to study the gene function in planta. It also allows reliable silencing of genes within a short period of time. We don't need to create the transgenic, therefore it is a routine process that can be completed within 1 month. There are wide range of procedures that are available for genome inoculations, like agro infiltration that we are doing by the syringe, then agrodrench, then leaf rubbing, and we can also use the biologistic, but mostly we use the agro infiltration. It is the robust methods. Robust protocols are available for different plants to carry out the VIGS experiment. We can easily know the function of those genes which are essential for the plant growth and development, that cannot be knocked out for their study, and large scale application of these experiments are possible, and we can compare gene functions between different species with the same constructs, and sometimes it is a heritable, the gene silencing heritable, because DNA methylation can be carried out into next generation. So what are the challenges of the virus-induced gene silencing? The first and foremost challenge is the identification of suitable virus vector and inoculation method in non-model plants. In model plants, it is already available, so we are doing the experiments in model plants, but for non-model plants, we have to identify the virus vectors as well as inoculation methods. Like for monopod, it is problematic. We have to use different monopod viruses for that. Okay. The other challenge is low level of silencing, because of some gene controls by different metabolic pathway. So if we silence that gene, sometimes what happens, the metabolic alterations will lead to the compensation of that particular transcript, so we cannot observe the particular phenotype that we are expecting. Then there are also functional redundancies between gene family members. So if we are silencing one gene family member, the other gene family member will take their place for performing the function. Then sometimes the symptoms that is caused by the virus-induced gene silencing that can mask the phenotypes that we are expecting. Okay, and the main disadvantage is it is the transient method. It is not permanent. After 1 or 2 months, the plant will recover. Then specific silencing of isoforms. Like if one gene is having many isoforms, so there will be difficulty to identify the unique region in that particular gene. So therefore, those genes cannot be studied by the virus-induced gene silencing. Then metabolic rerouting that we already seen for different biosynthetic pathway, different for one product, different pathways can contribute. So if we have to silence that gene, we have to this VIGS cannot be used for that. Okay. So this was the lecture for RNAi and virus-induced gene silencing. I hope you understand the lecture, and in next lecture we will discuss few questions that is frequently asked for the RNAi as well as virus-induced gene silencing. Important questions that I can give to you. For RNAi, remember the discovery, the scientist, the components like DCL, what is DCL, what is dicer, what is Argonaute, what is RISC, that you have to remember. Also, now they are asking which DCL is giving rise to 21 nucleotide siRNA. Then they ask siRNA, difference between siRNA or miRNA, and they are also asking transcriptional gene silencing, post-transcriptional gene silencing difference, and what is translational repression that is happening before the translation, because mRNA is captured by the duplex that we are having. Okay, and in VIGS they are asking why we are using the VIGS. It is a functional characterization tool like RNAi, but it is a transient and super efficient method, super easy method. Okay, so a reverse genetics tool, that is important, and because it is a virus-induced gene silencing, so we have to use the viral vector and we have to clone the specific sequence. So some questions are also asked about the advantage, disadvantage of the same that we already discussed. So where we can apply the virus-induced gene silencing and where this gene silencing cannot happen. So these are some important questions. Thank you for taking your time.